then they can pay. Okay? So we are trying to manipulate this gene such that we temporarily silence this, so and then create support the process for recombination. I hope some of you know that process. If you don't, that's okay. But I think you should have taken enough genetics and biology to know what recombination is. Okay, so how we are doing that and why? So it's what I just talked about. If we can put the entire right chromosome into wheat, but that will not be good because rye doesn't look anything like wheat, it doesn't yield as much as wheat, and it doesn't make nice enough chapati like wheat. So we don't want too many genes from rye, we only want useful ones. So we don't want this situation, like the entire chromosome, we want only this situation, that only small segment gets transferred and nothing else. Okay? So how are we doing this? I don't want to go through this detail, but we use all sort of system, and there are bioinformatics, your teacher who teaches bioinformatics, and I'm sure some of you are taking bioinformatics course. And so what is bioinformatics? I need a good answer, set of couple of good answers. Who want to define bioinformatics? Come on, don't be shy. Anyone? Seems like you want to say. Okay, so go on. You, yeah. Yeah. So what do you think? You can keep sitting, that's okay. Yeah, so what do you think? What's bioinformatics? Okay. So that is a lot of useful information. Who else? Yes.
So, application of information technology into biology to get a useful and desired results regarding experiments and all those.
Yes. Yeah. And that adds to the answer a lot. So that's very good. Yeah. And you can keep sitting while answering. You don't need to stand. So bioinformatics, if you go to, I'm pretty sure you will go through NCBI site or other databases where you do searches. Those databases are growing in information at exponential rate or even faster, right? So there's tremendous amount of information out there that you can get out if you know what you're doing. Lots of time, students or even PhD students, faculty think, oh, no, I'm not good at computers. You don't need to be good at computers. That's the beauty of bioinformatics. In fact, you don't need to be good at typing for that matter. You don't need programming. So, yes, if you are handling the computer science part of bioinformatics, you need some of those tools. But if you are a user, then you absolutely do not need that. Okay, then what do you need? High-speed internet to access those databases, and lots of clear thinking and intuitive searches. Okay, that's all you need. And internationally, the world recognizes Indian brain's ability in that area. Okay, so most people in the world admits that for bioinformatics, for putting 2 and 2 together, Indian brain works the best. Okay, all of you can be the best bioinformatician in the world just by, if you just cut down some of your Facebook time and some of your chat time. That's all you need to type. Nothing much, right? Nothing good will come out of that. Maybe some, but not much. But out of bioinformatics, a lot of good things will come out. Okay, so, and that's again very discouraging in this, like for me to see that India has not invested anything in bioinformatics. Yes, there are some bioinformatics programs, but you pick any bioinformatics journal, very few papers out of India, right? And how do you explain that? You don't need lab setup, you don't need a lot of money. So think about those things, right? And here, what we are trying to do, I'm not going to go into detail, we are trying to use rice information because rice genome is sequenced, whereas wheat genome is not. Okay, wheat genome is really large. It will be sequenced soon, but that sequence is not available yet. So what we are trying, so our objective was, we need to clone this gene first, right? This PH gene that some people call policeman gene, that it doesn't allow adultery among chromosomes. So then what we are trying to do is clone that first, and then we use it to control recombination ourselves. And then, so there, what this figure is showing, we took the wheat chromosome markers, DNA markers. If you don't know what those are, that's okay. And aligned that what part of rice we need to access. And this was all done by bioinformatics. And this colors is only showing, and this is wheat chromosome. This is busy slide, you don't see it, it's okay. But you can easily see colors. So what does these same colors mean? Same region in rice. So that means more of the same color in a specific region of wheat means very high homology between wheat and rice. So that means we can use rice sequence to address wheat problems. Okay? So one thing you can say is, well, wheat is not sequenced, I can't do anything useful. Other is, well, let's see what we can do to solve the problem. So we chose rice route to get to this gene. Okay? And we came up with this situation. Our gene was in this region, and it matched with one chromosome on rice, chromosome number 9. Yes, there was a small segment from another rice chromosome, but most of the markers matched with one rice chromosome. So, and this was all a wonderful example of bioinformatics. So the student, Gagan Sidhu, she is actually now just finished PhD at Cornell after this was her master's project. She probably spent a few months sitting on the computer non-stop trying to go through rice, and that's how you can clone genes easily. Otherwise, in wheat, if you want to clone a gene by other means, it still takes 10 to 12 or even 15 years.
So what is the size of this gene Th1? Is it a split gene or
it's a single gene, pretty small, about 500-600 amino acids long, not very long, and it has a couple of very interesting domains. I'll show you.
Around 80 kilodalton. 70 to 80 kilodalton.
You will know that better.
Yes.
Yeah. Okay. So what we did, so this was what Gagan came up with, that she identified about 93 genes from rice, and she wanted to know if one of these genes are the PH gene. Okay, we are talking about an MS student, okay, who is doing a main project, doing something else, and doing this as curiosity. Okay, and so once she narrowed down to 93 genes, so then the problem started, right? What do you do with the 93 genes? How do you test 93 genes for your topic? And that's where we invested in this, okay? So in humans, if you read some papers or read some medical press, then you will, and I recommend all of you reading some of the medical press. It is extremely exciting, even for not anything else, just for your information, what's going on in the world. You should read some of that, and go to WebMD on a regular basis. You will never regret doing that. Anyway, so in clinical 2 trials, there are many viral vectors being used for gene therapy. Okay, so what I mean by that is, Right now, the medicine, what we practice, is not curing, actually fixing the problem temporarily, right? We have no cures, right? What do we do if we develop heart problem? We just do plumbing, right? We just put a catheter tube in, open the blockage, right? Angioplasty, and but do we know why those plaques form? No, we don't, right? Do we know which gene make you more vulnerable to that? We didn't until now, but we will eventually, and some of those genes are known now, okay? So, we develop some other ailment, then we don't know why that's happening. For the most part, we don't know that, but we fix that to make you feel better, that's all. And but the future medicine is gene therapy. And scientists are finding out, as I talked earlier, that there are specific genes or proteins that start to malfunction. And then because of that, you develop certain diseases, okay? This aging process, I am oversimplifying, but that's okay for now. That one protein, which is in full form, it stays good until the age of between 30 to 35, and then it loses its proprotein, the first part of its gene. And many scientists believe, not all, there's a controversy still, many believe that one protein malfunction mess everything up and basically triggers the whole aging process, okay? The modified form of that gene was put in C. elegans, and those C. elegans their story was actually again on NPR, at the radio, that does a very good job in the US talking about these issues. So that lady, the scientist who did that, she said that those are still looking very strong. I think it's what 200 some days now. Yeah. So once you identify those genes, how are you going to take the good genes to the tissue? Let's say that bad gene is functioning in kidney and causing some kidney-related problems. Okay, so scientists are making kidney-specific vectors, okay, and taking the good gene to that kidney. Okay, what are those vectors? Mostly viral vectors.
And it will infect all the cells.
Right. Yeah. So, well, even some, so they can produce enough good protein there to counter the bad one. So there are many viral vectors that are in stage 2 clinical trials for gene therapy.
And I think all the mammalian cells, this is the only way. One is this, because they have the ability to spread us.
Right, absolutely. So they have evolved over millions of years, and we can just put them to use for our benefit. So we are trying to do something very similar in wheat. We are not trying to solve any disease problems yet, but we are at this stage of understanding gene function. So what we wanted to do was that we wanted to test those 93 genes, see if any of those are genes of our interest. So then we use this system. This is a barless peak mosaic virus, and it is RNA virus. It has three components: alpha, beta, gamma, and we put our gene in RNAi format. Okay, who knows what is RNAi? You better know that. Okay, speak up. Seems like you know what's RNA. No? Okay, you know RNA. Okay, what is it? Okay, very good. That's interference RNA. So how does that work? Okay, so there are many double-stranded RNA viruses, and to protect organisms against those, eukaryotes developed this system that not to ever tolerate double-stranded RNA. Anytime there is double-stranded RNA, there is a common enzyme that detects that double-stranded RNA, binds to it, and then this what's called dicer breaks down the entire RNA molecule. Okay, extremely efficient and slick system, present in all eukaryote studies so far. Okay, so we made artificial double-stranded RNA. So what we did, that all those 93 genes, we made antisense of those, that is complementary strand. I saw a beautiful picture of DNA there, so I'm sure you all know what's complementary strand. So that when this virus takes our RNA into the cell, it forms double-stranded, and then the dicer molecule chops it up, and then our gene is destroyed, and then see if we see any phenotype. So what we wanted to see was that any of our target genes, if it is PH gene, we will see change in chromosome pairing. The chromosomes that normally would not pair will start pairing. That's what we wanted to do. So these are just a bunch of tests we did. I'll just get to the point. So this is normally what that PH gene will do. This is one of the wild relatives, critical peregrinem, and this has actually many useful disease resistance genes that we can transfer to wheat. And but if these chromosomes don't pair, you can't transfer anything. But you remove the PH gene, then they start to pair. So that we had a pretty good system to score our phenotype. Let's get to, okay. And normally, this is what you see. Got this gene, which we believe is the PH gene. And now, we are trying to put this gene into action. That is, we are making the first crosses of wheat with rye, because we want to transfer heat tolerance. And we are applying for grants multiple places, including India, because heat tolerance is a huge problem in this region, especially this region and all the way to Madhya Pradesh. We lose up to 20 to 30% of wheat crop every year, without exception. Sometimes we lose 10%, and imagine what 10% yield would mean. Any one of you, if you can start wheat transformation facility on a service basis, given you business ideas, you will be extremely successful. You can produce plants for the world, because such facilities do not exist anywhere in the world. Okay? All you need is a small growth chamber, a laminar flow hood, and lots of good hours put into it, right? And you can charge good money, because not too many people can do it. Okay? So here we ask agrobacterium to take our gene into wheat plant. What is agrobacterium? Okay, it's bacteria. What does it do?
It transfers the gene of interest into plant.
Right, okay. So many of you have read that it has ability to transfer DNA into plant DNA. So we use this system to transfer genes into wheat plant, and it works really good. And then in about 6 to 8 weeks, we get these plants going through this process, and they will carry the gene of our interest. And then in this case, we wanted to silence a gene. This is normal expression, and then in our transgenics, the expression did not come. Okay? So this is just one of the examples that you can use a combination of different approaches, bioinformatics, some molecular biology, some cytogenetics, and then you can solve the problem. So now we are preparing a big project where we are proposing that until GMO wheat is accepted in the market, which may be 5 years or which may be 50 years, we don't know, no one knows, right? Even though every single one of you have eaten GMO product by now, I can say it without knowing what you eat, right? So it is that common that every single one of you have eaten some sort of GMO product in your lifetime. There is still lots of resistance to GMO. So we are setting this procedure up based on the PH gene to transfer useful genes from wild relatives of wheat into cultivated wheat, and without going through the GMO route, and just by simple crossing and manipulating a single gene. Okay? Or how are we doing with time? Okay. So questions. Okay? Before we go on with the next example. Yeah. Go on.
This is safe, as we know, Pt-cotton is not yet resistant to the bollworms.
Okay, so you are worried that the Bt cotton will create.
So Pt-cotton, when it was introduced in India, in Gujarat,
right.
so it was effective for all the bollworms.
Right.
But at this time, there are many bollworms which are causing problems.
Right.
Not dying spiraling.
Right. Okay.